continuation plasma membrane. One thing, it is also known as cell membrane, and it is also known as plasma lemma. All are the same, any name you can use. Now, yesterday we have studied that it is a selective membrane. Means it allows to go some things through it and some things cannot go through it. That's why it is known as selective membrane, and we have studied about it in last video. Now the next one is some substances like CO2 and O2 also go through it by the process diffusion. Means by means of diffusion the CO2 and O2 also come across the plasma lemma. Now what is diffusion? You should know. Diffusion means transfer of molecules from higher concentration to lower concentration that transfer molecules from high concentration to lower concentration until equilibrium. Means the molecules can go from high concentration to low concentration. This process is known as diffusion. Now, suppose that this is the cell, it is the external medium. If the oxygen concentration is high, if the concentration of oxygen is high, then inside the cell, then oxygen will enter. Means from high concentration to CO2. And similarly, in case of CO2, if the CO2 concentration is greater than the atmospheric concentration, then CO2 will enter. Thus, exchange of gases takes place through the cells by the process diffusion. And so thus we can say that transfer of molecules from high concentration to low concentration until the equilibrium is called diffusion. Now for transfer of water molecules, there is also a process that is known as osmosis. Osmosis means transfer of water molecules from higher water concentration medium to lower water concentration medium means that is transfer of water molecules from high water concentration molecules medium to lower concentration medium means from dilute to concentrate, from dilute solution to the concentrate solution means water will go from dilute solution to concentrate solution. For this an experiment is also we do that. This is the beaker containing water, and next we insert a tube. It's containing a membrane that is known as semi permeable means this phenomenon takes place only they are separated by a semi permeable membrane. So we can write when they are separated by a semi permeable now in this sugar solution. Keep this apparatus for some times, then you will see that the level of water has increased in the tube. That means water has entered to that, means the concentration of water is greater in outside and low in the inside. So from high water concentration, the water enters the low concentration medium, and when they are separated by a semi-permeable membrane, and that process is known as osmosis. So osmosis is a process in which the water molecules transfers from high water concentrations to the low water concentrations. And so now we shall discuss about its importance. Now suppose it is a cell and that is kept under a bucket containing a dilute solution, containing a medium. This is the external medium, and inside the cell, that is the cytoplasm, that is the internal medium. Here is also solution is present. It's water concentration, and it is also water concentration. Water concentration of internal site and water concentration of external medium. This is now if the water concentration of internal medium is lower than the water concentration of external medium, means water concentration of internal is less than water concentration of external. Then water concentration, we know that water molecules moves from high water concentration to low water concentration. So the water will move to the cell. Water will move to the cell. If it is greater, it will move to the cell. It means water will move towards the cell. So the cell bulges, and that is called hypotonic. That is called hypotonic. Hypo means below, means the concentration of inside is below, water concentration of inside is below than the water concentration of the outer medium. If the water concentration of internal site is same the water concentration of external medium, means WCI, water concentration of internal, is same to the water concentration of external, then there is no change. Means water will not flow from any sides. That is known as isotonic. Iso means identical, means both mediums are same in concentration. Next, if the if the water concentration of internal is greater than the water concentration of external, means WCI is greater than WCE, then the water will move from cells to the external medium, and that's why the cell will shrink, and this is called Hypertonic means hyper means above. When the cell concentration or water concentration in the cell is high than the water concentration of the external medium. So that's why by comparing this concentration we can tell whether the condition is hypotonic or isotonic or hypertonic. In one thing in such case means in this hypertonic, in such case the protoplasm shrinks, so it is also called plasmolysis, means breakdown of protoplasm. In this way, here the cell will bulge, and cell will bulge. Here there is no change, and here the cell will squeezed. Cell will squeezed. So in such conditions, either maybe hypotonic, isotonic. Hypo means below, if the concentration inside the cell is below, if the concentration of both sides is same, and if the concentration of the inside, water concentration of the inside is high. So such situations may be hypotonic, isotonic and hypertonic. And in this condition, in the hypertonic, the protoplasm get shrink, and they get squeezed. That's why it is also called plasmolysis. In biology, we study about plants and animals. The study of plant is called botany, and the study of animals called zoology. So the cells are also of two types. One which is found in plant, it is known as plant cell, and one which is found in animal, that is known as animal cell. One things, draw the diagram from your book. Now the difference between plant cell and animal cell. First one is cell wall is present in plant and absent in animal cell. Next one is plastid, it is present and is absent in animal cell. Due to presence of plastid, the plant make their food by own, which we will study in the next classes. Next one, backwards, large, a small or absent. Next one is very less in number, and here is more in number. Here is found in, it is found in greater amount and it is found in low amount. Next one is lysosomes. centrosome absent, present. So these are the difference between the plant cell and other cells, and other differences you may see from your books. Now we have studied that the central part of the cell is called nucleus. So on the basis of development of nucleus, the cells may be prokaryotes and eukaryotes. Means primitive nucleus, that is incipient nucleus, because the nuclear membrane is absent. Nucleus is not bounded by any membrane. That's why it is called incipient, because all the nuclear contents of the nucleus are scattered, while in the eukaryotes it is surrounded by the membrane. Next, membrane bound celled organs are absent, like for mitochondria, ribosome, Golgi bodies. They all are absent and these are present. Now ribosome is of 70s type, and here is ribosome of 80s type. It is composed of 50s and 30s, while 60s plus 40s. Now you will confuse that how 50 plus 30, 80, but 70. Suppose this is a of 3 inch and this is also 3 inch. Combinedly they are 6 inch, but when they will bind to each other, the length will be decreased. So in such positions they are present, means like that. Means 60, 40, but the total is 70 from this to this. And in the same type there is a TS 60 and 40, understand? And the 60 is so this is 60s, this is 40, but total here is last to first, that is of 80s.